Gary Finnegan from Preclinical. A question for Darnisha. You put up a slide there quickly about the kind of time frame. Could you basically speak that slide? Because I missed it.
Yes.
Just tell me what's coming.
Thank you. So we are actually entering into a preclinical safety tox program late this quarter, and we will actually seek to move right into compassionate use trials. So we'll take our safety in vivo data with our in vitro efficacy data and move right into human clinical trials. Again, a same pattern, a same platform and plan that was followed by Fusion T20. And those timelines for us can be as early as within 12 months, given the global crisis, maximum of about 24 months. And again, given the global crisis, and we feel like that is a potentially very significant to bring hope to what is happening and a real solution that could have a very immediate impact with the flaviviruses, specifically Zika, at this time. And I can share the exact slides so you can see all the breakdown.
My name is Richard Anthony, working in Amsterdam, and I had a question for Joe Turner. You were talking about targeting the symbiotic bacteria within the organism. I come from a tuberculosis background, so we are obsessed with multi-drug therapy. That would seem like an ideal opportunity to combine the drugs. Probably this is not my area, so it's already been done already, but maybe you'd like to say something about that.
Yeah, certainly. So that was one of the objectives of AWOL, was to look at antibiotic combinations. And so we started out in 2012 with a preclinical program looking at combining rifampicin with either doxycycline or minocycline, and combinations potentially of moxifloxacin with either rifampicin or the tetracyclines. What we found, what we quickly discovered, however, which kind of rendered that line of research, deprioritized it, was that if we just simply elevated the dose of rifampicin to within the newly defined safety margins that's been described for TB, so clinical TB. So you can elevate now rifampicin dosages threefold to fourfold, and this has been given to TB patients for upwards of 6 months. So when we did the mouse to human PK bridging and emulated those sort of exposure levels in the mouse model, we got a very rapid decline in Wolbachia. So in 7 days, in fact, in our primary screen, and this sort of trumped anything we could achieve with standard dosages of drug combinations. So for that reason, the combinations were parked. However, we are now considering combinations of antibiotics with standard antifilarial drugs, and we're very interested in combining rifampicin or our lead new chemical entity, the 16-membered macrolide tylosin, with albendazole. And we have some preliminary data to suggest that combining those has some synergistic effects in more rapidly targeting the embryogenesis pathway of the nematode. So that has some benefit in embryogenesis blockade of transmission, and also limiting the disease of onchocerciasis, because disease is caused by the microphylodermic stage in the skin. So yes, combinations are on the cards, and we're obviously underpinning that with PK/PD modelling to understand whether we're getting real synergy or additive benefits of combining classes of drug.
And for avoiding resistance as well.
Yeah.
Which you would not see
Yeah, so I mean, yeah, certainly. I mean, resistance is another area that we're actively pursuing more in the cell line work. So we're trying to drive resistance to monotherapies to understand when and how that is achievable, and also to understand the targets involved, especially with the Tylomac, because the Tylomacs are so potent. We may, whilst putatively it's a ribosomal target, there may be actually other targets, off-target effects, yeah.
Thank you. There's a question to Dr. Pearce. I can't see you behind me. You might have mentioned it, but what's the difference in the structure, kinetics, and also phenotypic activity of your compounds, looking at the three different main species of schistosoma? Mansonae, hematobium, and...
We've only tested them against schistosoma mansonae at the moment.
So have you got target of the two other parasites?
Well, what I can say is that in terms of histone deacetylase 8, all the schistosomes have the same configuration of their catalytic pocket. They all have, it's highly conserved between the schistosomes. In other words, they all have the histidine, they all have the flipping out of the phenylalanine. So
it looks
theoretically, anyway, anything we do will be applicable.
My question was to Alan. So, looking into a panactive compound within the nitroaromatic compounds as FEXI, what can you comment on the different NTRs or mechanism of activation across the different kinetoplastids, and also about to overcome the toxicity in the host, in the human being? So are there any other mechanism of activation or reduction of? Okay.
So delaminid and the other bicyclic nitro drugs such as PA824 and VL2098 have biological activity against leishmaniasis and against T. cruzi. They have very weak activity against the African trypanosome. And the reason for this is that the African trypanosome does not contain an ortholog of NTR2, only the oxygen-insensitive type 1 NTR1. So clearly there's always been an issue about the safety of nitro compounds, and indeed led to the abandonment of a development project when I was in WHO TDR in the late 80s or early 90s, MK436. There were clearly serious concerns about the potential toxicity and mutagenicity. But I think the key thing is, if these compounds are specifically activated by a unique target in a bacterium, such as in Mycobacteria, for example, it's DDN that activates these bicyclic nitro drugs, or NTR2 in Leishmania, and that is completely absent from the host, then part of those toxicity issues can be ameliorated. But I think in any process of drug discovery, there are issues. If you're targeting kinases, then you have to worry about inadvertent inhibition of the 500 or so human kinases. If you work on HDACs, histone deacetylases, then you have to be concerned about cross-inhibition of a human enzyme. So finding unique targets is very rare, and therefore we should exploit them whenever possible. We've made some of the metabolites that are formed, and some have weak activity against the Leishmania parasite, as is the case in TB. But I don't see them as perfect development candidates because they're just not potent enough. So we're still stuck with having that nitro group on the molecule. And I understand perfectly well why all medicinal chemists have been immunized against nitro groups over the years, and that's why an academic working with a chemist is able to make a contribution that otherwise might not be possible.
Thank you.
Okay.
This question is for Darnisha. I apologize if you mentioned this and it went over my head, but are there any challenges, mechanistically speaking, with regards to Zika that are unique to that compared with dengue? Is there something about the virus that you see differently from that regard?
So I think I probably did not address that, and that's a good question. There's so much still about Zika that we don't know, but what we are foreseeing are some of the same and very similar responses that we see with dengue that has been far much more and better understood. So there's so much we don't know, yet still we are truly believing that there will be very similar responses and to the targets. Now, with antiviral drugs, the most effective with peptides have been historically with combination therapy. The very good news for us is that under our proprietary platform, we've got 4 peptides and with 2 very distinct mechanisms of action in inhibiting binding as well as fusion, which are very highly conserved processes in viral replication within the flaviviruses. So we feel very confident because of also the highly conserved nature of our peptides, of that synergistic and combination therapeutic effect. So, and we won't know this for sure until we do get into the clinic, but lessening disease progression and thereby by helping to lessen the spread, we think will be significant.
Hi. This is very much out of my field, but I'm just curious that you're working with leishmaniasis, and I'm wondering if there's any strategies to develop like a vaccine for leishmaniasis. Is it possible?
Well, I suppose I can comment on that. Yeah, I mean, there have been the concept of live attenuated vaccines for a number of years, but I think they're still very much in the preclinical development. The problem with Chagas disease and with leishmaniasis is that they are hiding inside a host cell, and therefore it becomes more difficult to develop vaccines. But there have been DNA vaccines, live attenuated vaccines, combinations of proteins, including one of the trypanothione-dependent enzymes I've worked on. So I think there's a range of different options, but unless Simon, the expert, or Karen maybe.
Yes, if I can just comment. So there are a few vaccine candidates which are actually in development currently. There is one vaccine candidate by the University of York, which is in clinical development. As far as I'm aware, they have completed phase 1 and are moving forward. They have other clinical vaccine candidates being developed in the past few years. One of which LSHDM and other partners were involved, funded by the European Community, that would kind of be ready to go into clinical development, but there has not been further funding so far. So there are definitely efforts, and there are definitely vaccine candidates there at the moment, some of which are moving through the clinical development.
By the way, there are in veterinary leishmaniasis, there are already some vaccines which are more advanced. Efficacy is debatable. That's another issue. But the first vaccine that were commercialized were in in the veterinary domain. You must know it's around the Mediterranean basin and in Latin America, you have visceral leishmaniasis where the dog is the reservoir and is suffering a lot. And so that was the first target of these first vaccines.
I think we've got time for one question more, and then we're going to break for lunch. So,
hi, I'm Mo Alavija from Pharmadex. I was wondering if the panel could comment on the key technology limitation which you sort of facing, or key platform technologies that could help you to fast forward your drug discovery. Would it be a sort of super sensitive, more penetrative imaging, or would it be more selective proteomic type technology?
I'll start. Well, the drug discovery unit in Dundee is fully integrated in everything except toxicology. We do all our in-house models and pharmacology. And I think the challenge still is the animal models, and most particularly in the case of Chagas disease. And really the thing that's driving that model forward is John Kelly in the middle of the audience there. And of course, with Chagas disease, we don't actually know, if you remove the parasite, whether the disease progression will be halted. According to the BENEFIT trial, where they treated with benznidazole and apparently removed parasitemia as best as we can measure it in blood samples, there was no benefit to the patients in long term in terms of cardiac symptoms. So there are clinical issues downstream which we don't understand, and definitely we all need better animal models. And I was delighted to hear about the new Helminth models, which have bedeviled progress in that field. And no doubt that'll be commented on.
Yeah, I'll pick up the baton.
Yeah.
I think preclinically, certainly we need more rapid prognostic indicators of drug efficacy, because the preclinical models require quite a sustained period of time to assess death of worms, even for direct-acting agents. And so it's something we are looking to develop in terms of ultrasound to detect motility, and we have some evidence that we can use ultrasound prognostically, so we can get a more rapid handle on loss of motility of parasites in the animal. And we're looking at bioimaging, other bioimaging modalities to hopefully accelerate the time frame to get the data back for decision making as to next steps. But I think more problematic is going into the clinic for onchocerciasis, because we don't have a validated biomarker for adult living onchocerca. And obviously you can cut out the nodules, and you can do nodulectomies, which is a very invasive procedure to assess viability of the worms. You can use surrogates such as the MF in the skin to determine when that falls away, which again is an invasive procedure. So there's a real shortfall of more rapid diagnostics, less invasive diagnostics, to determine when a drug has worked in an earlier, more accelerated time frame.